Hi, in this lecture, we are going to finish up our study of renal physiology and how urine is formed by looking at the remaining two processes of reabsorption and secretion. So remember that there's three processes that have to happen for the formation of urine. The very first one happened up here in the renal corpuscle, and that's when we used our pressure to force material out of the blood into the nephron. Once it's in the nephron, we call it filtrate. So when we look at the nephron, it was that renal corpuscle, the entire structure, the blood in the glomerulus and glomerular capsule of the nephron was the renal corpuscle. The specific portion of the nephron involved in filtration was the Bowman's or glomerular capsule. Now the filtrate enters, and as it passes through the nephron, it's further processed because that filtrate, it's non-selective. It has a lot of good stuff in it. So reabsorption is when you move materials from the filtrate back into the blood. So think of it as trying to hold on to the good stuff so you don't lose it as urine. Secretion, on the other hand, is moving materials from the blood back into the nephron. So you're trying to get rid of these materials. And so we're going to look at reabsorption and secretion next. So reabsorption is important because the filtrate's very nutrient rich in the PCT. It's very similar to plasma. It won't have plasma proteins, but it does have a lot of good things like amino acids and glucose and electrolytes. Urine, on the other hand, is mostly water and waste products. So we need to get stuff from the filtrate back in the blood so it doesn't leave our body as urine. And that's what's going to happen through the process of reabsorption. Now, there's a couple ways of moving things. We're moving, through cells. So paracellular and transcellular are two terms that are used to describe this. Paracellular means that the materials move between the cells, whereas trans, it has to go through the cells. And most things are using this transcellular process. Terms to remember: osmosis is the movement of water. Water typically moves through little pores called aquaporins, and water has to follow osmotic gradients. It has to follow osmotic pressure and go wherever you have the highest osmotic pressure. That's what will pull the water. Diffusion, on the other hand, is just moving material from a high to a low concentration. Both of these are passive, meaning they do not require ATP. The cell doesn't have to use ATP to move the molecule. We have carrier-mediated. You've learned this as facilitated diffusion. So what happens here is you have a protein molecule and the substance has to bind to the transporter to be moved across. If you have more molecules than transporters, you don't get to move them all across. So you can actually saturate those transporters, and this is called your transport maximum, or we call it the renal threshold. And this is really important because glucose is absorbed through facilitated diffusion, and the renal threshold for glucose is about 180 milligrams per deciliter. So this is if your plasma glucose, this is the level in your blood, because if you have a lot in your blood, you're going to have more in your filtrate. Think of it as spilling over into the filtrate, and then you won't be able to pull it all back through reabsorption. So if your blood glucose is greater than 180, you're going to have more glucose molecules than transporters, and you won't be able to get them all back. So glucose will leave in the urine, and we call this glycosuria. When we look at active transport, we have a couple things that can happen. We can have co-transport, which is known as symport. So this is when you have two molecules, they bind to the receptor and they go together. Or you can have antiport, which means they go in opposite directions. A lot of our active transport mechanisms are typically going to use a sodium with something else. In fact, you're going to notice there's going to be sodium transporters. There will be co-transporters and counter-transporters. A lot of them are counter-transporters where sodium goes one way, and often we have this counter-transport with potassium or hydrogen. We have another one with sodium, where sodium can go, and then it can either come back with a bicarb or a chloride, and we'll be looking at these shortly. So when materials are moving, they typically are going to be going through the cell, and what you see here is a section of a PCT. You see the filtrate, you see the cell of the nephron, the simple cuboidal with the little microvilli, and you see the blood. We're not going to get too carried away and involved with how things move, but I want you to understand that it's a little more complicated than how I'm teaching it. You have to move it in the cell, out the back of the cell, and then into the blood. So they technically go through 3 layers. And I thought it might be easier to just make a table because reabsorption happens in multiple areas of the nephron. And if we look at reabsorption, if we take the nephron, notice you can see the reabsorption and you can see the secretion. And then the other arrows, the red and the blue, are just trying to represent if it's active or passive. So if we look at the PCT, this is most involved with reabsorption. So this is where 100% of your glucose and amino acids should be absorbed here, as long as you don't exceed the renal threshold. This is the only place we can pull glucose and amino acids back. We do reabsorb some bicarb here, in fact, a lot of bicarb, and water. Roughly 65% of the water is reabsorbed here as well. This is where we reabsorb the majority of our electrolytes, nutrients. This is from your textbook, trying to show you the proximal tubule, and just generally electrolytes. We're looking at water. As far as sodium goes, we usually reabsorb approximately 75% of the sodium is reabsorbed here. So the PCT is most involved with reabsorption. Now below that you see, how are things entering the tubule cell? Well, what I want you to understand is it uses a lot of transport with sodium. So glucose and amino acids actually are co-transported into the cell. So a sodium binds, a glucose binds, they get sucked in. Or you could make this glucose and amino acids. So we have to absorb sodium to absorb glucose and amino acids. Then they're going to be kicked out the back end of the cell. And as far as how they get in the blood, I just want you to know that the glucose and amino acids will use facilitated diffusion. Now, as far as water, remember that water is using the process of osmosis. So water will come across. As long as there's more solutes in the blood and the interstitial fluid, that will create that osmotic pressure, and that's going to pull the water out of the filtrate and allow the water to be reabsorbed. Now, if we look at the rest of the filtrate for a minute and look at what else is reabsorbed, in the descending limb, we reabsorb some water. And then we curve, and the ascending limb, we reabsorb salt. Remember, the descending limb was permeable to water, but impermeable to salt. And the ascending limb was opposite. It was permeable to salt and impermeable to water. In the DCT, this is where we can reabsorb sodium, we can reabsorb water. However, they're dependent on the presence of hormones. So in your notes, you have facultative water reabsorption. And what I want you to understand is that in order to reabsorb first the sodium, we need the hormone aldosterone. So typically we'll say roughly, depends, your book might see a slightly different number, but roughly 75% of the sodium is reabsorbed in the PCT. With aldosterone, that remaining 25% is reabsorbed because we excrete very little sodium in our urine. So the DCT will reabsorb sodium. It will leave the filtrate and enter the blood, but only if aldosterone is present. As far as the water goes in the DCT and then the collecting duct, water is only reabsorbed in the presence of ADH primarily. Now, ADH controls those little water channels we mentioned called aquaporins. So you can imagine all these little water channels, and ADH is going to open them or close them. So when we look at facultative water reabsorption, this is water reabsorption that happens only when ADH is released. And ADH is released a lot, by the way. Now, when we reabsorb sodium, a lot of times in the DCT, some water will follow sodium. And then we call that obligatory water reabsorption. It's kind of like obliged to follow the salt because it changes the osmolarity. In this image, it's a little busier from your textbook, but again, you can see the parts of the nephron, most reabsorption in the PCT, the DCT controlled by hormones. Aldosterone primarily affects sodium, and water will follow sodium out in the DCT. The collecting duct is mainly really dependent on ADH primarily. So we can say ADH mainly affects the collecting duct, whereas aldosterone is the chief hormone in this DCT as we're looking at these processes. Now, as we reabsorb water, so if we have a material that's in the filtrate and water leaves, it's going to concentrate that. And what happens is we do have in our filtrate, we do have some urea. And so if I have urea, but then a lot of water leaves, what that does is that increases the concentration of urea. Well, urea is lipid soluble. So what that means is it can pass through cell membranes. So now urea comes back in the blood. So we do reabsorb some urea. It's called solvent drag. And it happens because when water leaves the filtrate, it concentrates, right? This is more concentrated. So the urea concentration goes up, and then that gives it a gradient for it to come right across. So we do reabsorb some urea in our PCT. As far as the bicarb and the CO2 go, just understand, because this equation's coming back soon, that we have our CO2. It combines with water and forms carbonic acid, and then that carbonic acid breaks down into bicarb and hydrogen. So most of the time, this happens inside the tubule cell. The bicarb is going to try to go in our blood because we want to hold on to it, and then we usually kick the hydrogen out. And then this reaction can also happen out in the filtrate. So the bicarb and the hydrogen are mainly dependent on CO2 levels and this equation happening to generate bicarb and hydrogen. But we're going to look a little more specifically at them in just a minute, because really their reabsorption and secretion is mainly dependent on pH. And then we come back to our hormones. We had PTH and calcitonin, and they are really important in controlling the reabsorption of calcium. So calcium is mostly reabsorbed in the proximal convoluted tubule. So remember that calcitonin was the hormone that was released when we were having a high calcium level. And so calcitonin is going to decrease the reabsorption of calcium, whereas PTH was released if we didn't have enough calcium. So PTH is going to increase that reabsorption of calcium. And so this is the table you have to fill out in your handout. And so again, if going through and filling out, I think you've probably filled out most of it, but just remember that the descending limb is permeable to water. The ascending is your sodium and your chloride. So these are what are reabsorbed and where they're reabsorbed. We can just say the majority of the electrolytes. We know that glucose and amino acids are reabsorbed here. And then I'm just going to write about 75% of the salt. As far as the DCT in the collecting duct, this is where we have facultative and obligatory water reabsorption. Let me do the salt first. So the sodiums are reabsorbed if we have aldosterone. And aldosterone's target is the DCT. Water will tend to follow sodium when that happens, so it will cause a little bit of water to come back, and this is called obligatory. It's obliged to follow the salt. The collecting duct is really sensitive to ADH because ADH opens up those aquaporins. And if you have ADH, you reabsorb even more water, and that's facultative water reabsorption. We talked about the processes for how the materials move back. Water's always osmosis. As far as these salts, I'll just write it down here, they use active transport. They're actually pumped. And then I just wanted to review this with you and remind you that it's mainly due to the pH that's going to occur. And then we talked about the solvent drag, is that as the water leaves, it gets more concentrated in the filtrate, and therefore some of it leaves. So some stuff that's not reabsorbed, it might be because it's too big, like creatine. It might be that it exceeds the renal threshold. So maybe if you have too much glucose, that can also occur. Now, secretion's the opposite. Secretion is your body's last chance to get rid of something it doesn't want anymore. So the blood is actively secreting things back into the filtrate. This is how we get rid of a lot of waste. A lot of drugs are broken down and eliminated in our kidneys, or something that we might have reabsorbed we need to get rid of. And so when we're looking at secretion, Primarily, we're looking at the DCT. Okay, so you can see that some of it happens in different parts, but we will focus on the DCT. So why is it important? Well, like I said, you got to get rid of anything left over in the blood. When filtration happens, only 10% leaves. So there's probably more waste products in your blood. This is how we control potassium levels. We might have reabsorbed things that we didn't mean to previously, and it's the number one mechanism for controlling blood pH. So we have our filtrate, we have our tubule cell, and we have our blood. And in this case, we're moving things from the blood into the tubule cell to get rid of it. And as I mentioned, this is one of the main ways that we're going to control pH. And when we look at these transporters, we have a countertransporter with sodium. Now, it's either with a hydrogen or a potassium. So what that means is they have to compete to leave because they're going in opposite directions. So in this image, you see a sodium coming back to the blood. And then in the red, you see it's kicking out a hydrogen at the same time. So this is a sodium hydrogen countertransporter. And that transporter can either move a hydrogen or a potassium. And then we also have these transporters with bicarb. Now, what happens with bicarb is, again, we can move sodium, but bicarb competes with chloride because they both have a negative charge. So if we look at this, it's really dependent on the pH of the blood. So I have two tubule cells here. This, we'll just pretend that this is the filtrate out here. And so we'll do number 1 here and number 2 here. So we're going to have in our blood, we will have hydrogen, we will have potassium, and I'll use another color for the other ones. And the anions, I'll do this purple, pink thing. So what's going to happen is sodium is going to want to come back. We really want to hold on to as much sodium as we can. So the sodium is coming back to the blood. Now, when it comes back to the blood, either a hydrogen or potassium can go, and we'll just draw 2 sodium transporters to keep this simple. And the other transporter is transporting anions, so either a chloride or a bicarb. Okay, so let's say pH goes up. So if your blood pH is too high, let me get another color here. So blood pH is too high. If the pH is too high, that means that you don't have a lot of hydrogen ions in your blood. In fact, you probably have too many of those bicarbs. So what you're going to want to do if the pH is too high is you don't want to get rid of that hydrogen ion. You need to hold on to it, right? So what you're going to do is the potassium is going to be able to leave because you're holding on to hydrogen to bring your pH back where you want it. And then if we look at our base, well, you know, we have too much bicarb. Now we're going to want to get rid of the bicarb and we'll hold on to the chloride. So when pH goes up, you're going to want to hold on to hydrogen. So you're going to decrease the secretion of hydrogen, which means you're actually going to get rid of potassium. So you're going to increase potassium secretion, and at the same time, you're going to get rid of the bicarb. Okay, well, what if we have the opposite situation? So what if my pH falls? Okay, well, if my pH falls, now I know that I have too many hydrogen ions, right? I need to get rid of them. I have way too many of those. And then if I'm looking at my base, I'll put down here my negatives, my chloride. Well, if I'm too acidic, that means I don't have enough base. I'm going to need to hold on to that bicarb. So when my pH goes down, now I'm going to get rid of that hydrogen ion. Remember, sodium's coming back no matter what. So again, I'll just draw my little sodium here coming back. Same thing here. And in this case, though, when I'm trying to get rid of, if we're looking at the base, right? So if my pH goes down, I'm too acidic. Now I want to hold on to bicarb, so I'm going to kick the chloride out. And so all it is whichever ion goes is dependent on the pH. And then this definitely can cause imbalances in potassium. Notice sometimes we kick it out, so it might fall, or we hang on to it, it might go up. And that does happen. And we'll look more at that when we do fluid and electrolyte and acid-base balance. And then the last thing is also understand that aldosterone also controls potassium. So when aldosterone is released at the DCT, it's always going to bring sodium back. That's its job, to increase sodium reabsorption, but it activates a very specific countertransporter, and it always kicks potassium out. So aldosterone is always secreting potassium. We lose about 10 to 15% of our potassium every single day just through urination and aldosterone. So it's important to make sure that you're getting enough potassium in your diet. So this wraps up our lecture on reabsorption and secretion.